All right. If you are just joining, there's a poll everywhere. That's a survey to start this course. So go ahead and start that, and I'll kind of fill in and talk about what we're doing while that happens. All right. Let me get this chat going. Can someone put into the chat, can you see my chat and videos, or do you just see the PowerPoint? You should just see the PowerPoint. Awesome. Okay. So, as I've told you, this is my favorite lecture to give you guys. I've got you for 2 hours, and this is kind of like just an amazingly burgeoning field of knowledge and how we can increase our biological quality of life. So the giraffe here is because when evolution was first kind of being discovered, Lamarck, as opposed to Darwin, said he was wrong, but he said, you know, giraffes' necks are tall because they keep reaching for the leaves, and so their body trains them to go higher. So while that is wrong, there are epigenetic effects that kind of have that manifestation. So we do things in our bodies adapt to our environment. We're going to talk about, you know, how smoking affects your genome, how epigenome, how bad not getting sleep is. All the good stuff, you know, there's a lot of it has epigenetic mechanisms. Broccoli, how bad is alcohol or eating a lot of food. There's a lot of things that we'll talk about that the IRBs, the ethics boards, wouldn't allow studies to be done on humans for good reasons. And so a lot of times we have to look at natural disasters. So there's data on 9/11 or on World War II, famines, and we'll go through all that. I think I have, hold on a sec. I want to quickly show, oh, I don't have it. Okay, well, there's online notes just like normal. And again, just like normal, do not read into them more than you want. They're going to have extra information. And I'll come back to these survey results. I'll try to keep giving you time to answer them. The sound is crackling and cutting off sometimes. Not sure if it's a Wi-Fi problem. I'm not on Wi-Fi, so that's not it. Let me see if I can switch the sound to, I have something else plugged in. Audio speaker, microphone. I am using the speaker. Okay, I'll switch to the internal microphone. Okay, testing. Tell me if this is better or worse. Yeah, I'm actually, so Panopto failed to update on my computer, so I'm actually on T. Arbor's computer with that microphone, but I switched to the internal Mac microphone. Is this better or worse, this sound? Or the same? Someone in the chat box. Better. Okay, we'll leave it there, I think. Okay, so objectives, same as normal. You'll see there's a massive one here that has 17. Describe the epigenetic effects of environmental factors discussed. Food, endocrine disruptors, sleep, light, exercise. And if discussed, to what degree they affect cancer. Okay, so that's kind of the most actionable, and focus on those. So we'll have some slides that you've had before, and then we'll get into how they're actually manifested. By far, the kind of most well understood and I don't know if I'd say targetable, but anyway, one of the best ways to think about how the epigenome is controlled is through these methylations of these CG islands. They're repeating CGs, and the cytosines get methylated, okay? And you can think of the methyls, the CH3, you can think of that as like a bump on the DNA that prevents things from binding. And so basically, if the DNA is methylated, it is turned off, because things can't bind to it to create RNA. Okay, that's probably by far that and histone acetylation is the the ways you'll be tested most on the boards. Okay, so then you've seen this probably from Dr. Gardner, the chromatin structure, and these little purple things are the histones and the DNA's wrapping around. So remember, we talked about histone acetyltransferases, HATs, and histone deacetylases, HDACs. And the HATs put on the acetyl group down here, and that turns the gene on because it's taking away that positive charge of the lysine. And the positive charge of the lysine wraps the negative DNA, shutting it off. So this is the off DNA here, all bound up because transcription factors and things can't bind there. And when it's unraveled, then it's turned on. Okay, so HATs turn them on, HDACs turn the DNA off. The other thing that you'll hear about for drugs is HDAC inhibitors. So an HDAC would be turning off the gene. So an HDAC inhibitor would be keeping the gene on. Okay, put into the chat if you have any chat box, if you have any questions as I go. So, and we talked about this before, the methylation is different in that it's of the histones, it is on the tail, and I'll have that summary slide again, and that's just kind of what I would remember. There's also these long non-coding RNAs that we'll talk about, and I know whenever I'm in the lecture room, students are typing away as I mentioned this and crossing out a slide saying, I don't need to know this. The long non-coding RNAs I won't test on and the boards won't test on because they are too numerous to keep track of. So there are tens of thousands of long non-coding RNAs. There's more long non-coding RNAs than there are genes, and they can turn genes on and off. So there will in the future, there's not now really, but there will in the future be drugs that maybe deliver a long non-coding RNA that are known to increase or decrease that gene. And so, you'll learn that for that specific drug. But just know it's in a massive amount of regulation that we don't fully understand yet and we're not testing on. But I'll, of course, show you slides and data on it. Okay. So let's talk about 2 ways non-coding RNA stop protein production. And my spelling is awful. Okay, so you can block the promoter, right? So things can't bind there, turning the gene off. And then you can also have this RNA interference, and that binds via this RISC complex to block and destroy the mRNA. Okay, and so here this is a little old now, 5 years old, but this is showing you drugs and where they targeted. Using siRNA, which we'll talk about, and miRNA, which are types of RNA interference. So by far, not surprising, this blue one, 31% is cancer. Then interestingly, 21% were ophthalmology issues, but I think it's mainly because a lot of gene therapy early has been in the eye because it's easy to deliver. You can actually kind of just shoot things into the eye. And then genetic disorders is the other big green one. All right, so here are, so I have to keep moving the chat boxes and everything so I can see it. Here are, we're talking about miRNA and, well, let's just start with high level. Okay, so you have DNA, you have these histones, and you can methylate the histones and change the transcription. The DNA becomes RNA, becomes protein, and you have these miRNAs that can block or long non-coding RNAs. And remember, the long non-coding RNAs can do both, okay? On and off. So methylation of the histone at the promoter turns off the gene, and we're gonna talk about this RISC complex that blocks and destroys the mRNA. We'll come back to this figure and I'll kind of fill it in more. So here are the 2 RNAs that are used kind of as therapeutics to destroy other RNAs. And so here's siRNA on the left in red and miRNA in the right in purple. And the red doesn't mean anything here. There's a few kind of big take-homes I'll want you to get from this. The main one that's clinically relevant, I would say, is that siRNA is specific. So it will target kind of one mRNA. miRNA has multiple targets. And so if you look over at the right here, you have the number of patents in the US and Europe, and in red is siRNA and in blue is microRNA. And so these are for different diseases. You see the red bars are higher, and that's because they're more targeted, right? And so for miRNA, you would be a little worried about side effects because you're targeting other RNAs. Now, the other RNAs, when an miRNA targets a class of multiple RNAs, those multiple RNAs are similar because we'll talk about how they're hydrogen bonding, right, to have that specificity. So it could be in theory that like an inflammation pathway you're shutting down, and if you use an miRNA, it might shut down multiple inflammation mRNAs, and so that could kind of be a multi-drug, right? So one miRNA is really acting in a lot of places you want, and so it would have a better effect. But again, you don't have the fine-tuning ability as the siRNAs. So for both of them, So for siRNA, it's a double-stranded RNA, okay? So blue and red you see here. They both start off longer and then kind of get processed smaller. For miRNA, you have this hairpin loop structure early on, but it then gets cleaved, and so you end up again with a double-stranded RNA. And that processing for both of them, from the larger to the smaller RNA, happens through this protein dicer. I haven't read a manufacturer, like no one's taking siRNAs into miRNAs and you're having to worry about swamping dicer, right? I haven't read anything about worrying about the efficacy of dicer. That's just the mechanism it happens through. And then there's these proteins, RISC and AGO, that bind the RNAs in both of them. And the one strand I'll go through siRNA now, one strand in the siRNA gets cleaved off. So now you have a single RNA strand that can hydrogen bond. It binds to its target sequence and then signals for that mRNA to be degraded, okay? In a similar fashion, miRNA, the passenger strand is discarded, it goes down and it can bind to multiple RNAs that are similar, okay? So the take-homes, again, were siRNA, which is more specific to an mRNA. miRNA hits a few miRNA hits a few mRNAs, but they work through similar proteins. All right, I won't belabor this point. You've seen this slide on a version of this where on the y-axis it's kind of our DNA that's not coding for proteins, and humans have a ton of it because we use it to control things. Here's your table to show you what these things are. So miRNA is microRNA, siRNA is small interfering RNA, and that RISC protein is that RNA-induced silencing complex. So on the right here is that pathway I've shown you before, and just to show you how things can get complex, we've talked now about how miRNA and siRNA via the RISC complex can block an mRNA. Well, these miRNAs and siRNAs can also block long non-coding RNAs or other miRNAs. So you could inhibit an inhibitor, okay? Now, while that complexity exists, if you hear miRNA and siRNA, just think you're shutting down a gene, okay? Clearly, this is a big FYI slide. Here's the LNRNA slide. Long non-coding, LNC RNA slide showing you that it's very complex. I have a link there if you want, but since it goes up and down and there's so many, just know that you'll learn them case by case as they become therapeutics. Okay, I'm not going to do, I have a few poll everywheres that I'm not going to do. I'm just going to do that first one because the poll everywhere is so kludgy this time. There's a table I'll have later that answers this. You know, you should know for all these epigenetic mechanisms, what's causing it to go up or down? Is it turning on the mRNA, turning off the mRNA? And I just made the study slide that I would have made if I were you later on. So, and here it is. Okay. So, these are color-coded if they're going on and off. So we talked about DNA promoter methylation being the main one. The protein responsible for that are DNMTs, DNA methyltransferases, and they turn the gene off. Histone deacetylation, HDACs, turn the gene off. Histone acetylations, HATs, turn the gene on. There's histone phosphorylation, we haven't talked a lot about, and that's done by a kinase or a phosphatase, and that turns the gene on and can be used for DNA repair. Histone methylation via HMTs is mixed. It's more off the histone methylation, and you can also methylate a histone 1, 2, or 3 spots, and that differs the effect. So there's like this code of histone methylation we're learning. Again, along non-coding RNAs is mixed, and there's too many. It's complex. And then siRNA, miRNA, and then there's a thing called AsRNA. They work via RISC and AGO, and they all turn the gene off. Okay. Sorry if I miss a chat. Go ahead and put it in the chat box, but you can just say it too, because I keep having to move the chat box off to see what I'm doing. Okay. So unlike being sequenced, which in the next 5 to 10 years, I think patients will have that done because it'll be cheap enough. It's now $800 a genome or something, maybe 900. And once it gets to sub 500, we think it'll kind of just be done for everyone. But the epigenome is more expensive, and this is old again, 2015, but it's still more expensive. But if you're doing the whole epigenome, and there's different ways of doing it. So the gold standard is this bisulfate sequencing whole genome, but that's not really going to be done a lot. What could be done is kind of like how there's gene chips that measure certain genes. You could do it for certain, imagine an inflammation pathway of epigenetics, and that could be done eventually. If it gets cheap enough, it'll be done like at an annual checkup or something. Here, I told you to eat vegetables and start exercising, and then the patient comes in like, I did all that, and you're like, no, you didn't. I can tell from your epigenome that you didn't do any of that. But who knows when that will happen? Not on the near horizon. All right. So that was kind of some of the groundwork we had to get through. So we are going to start talking about at what level the epigenome can be changed, and how long does that change last, right? Does it change on an evolutionary time frame for more than 3 generations, 2 to 3 generations, just in your lifetime, for a few years, or per day? And long story short, it does all of those. Most of it is cut off at this pink and to the right. So 2 to 3 generations or shorter time frame. Okay, there's a little data on maybe over 3 generations, but mainly in animals and not really. And then I'll show you an example on an evolutionary time frame. Okay. Any questions? Whoops. Okay, moving on. So here is, I'll try to point out when I'm telling you stuff that is not board testable, but is me testable. So this is clearly not going to be tested on the boards, but I think it's pretty interesting and unique to, it is what humans are. So anyway, it turns out that once in a while, when during conception, you can duplicate an entire genome, okay? This happens in multiple organisms, it's just rare. So usually when you duplicate an entire genome, the fetus doesn't survive, right? But it turns out twice long ago, okay, about 500 million years ago, it happened to our ancestors. And so when that happens, you duplicate one genome, okay? And then that other genome gets epigenetically shut off. So it's kind of like it doesn't exist, and that's how you survive, okay? So that happened, and then it happened again and got shut off. So we had something like myoglobin, and then our genome got duplicated and we had 2 myoglobins. And then our genome got duplicated again and we had 4 myoglobins. One of those myoglobins became hemoglobin. One stayed as myoglobin, is it the current myoglobin? One became something like myoglobin, and then one got kicked out of our genome. That's what we currently have. So if you look at the graphs on the right, on the x-axis is going back in time for like how old that DNA is, and the long and short, again, not testable, but so you know what's going on in our body is, and on the y-axis is methylation. The left box is the promoter region, and the right box is the gene region. And so you can see when you're looking back, the gene region, the methylation, there's not really a pattern. It's the same in promoter. It's the same regardless of time. If you look in the promoter region, you can see that it started off methylated, and it kind of went away. And so long story short, you shut down the genome, and then over time they can get reincorporated, and those genes have changed, and so we have novel genes because of that. Not testable, but interesting to know for us. Okay, so how does it work in us really? Because that what I mentioned was just a kind of fluke that's not happening, in the thousands of years since, talking about the Ice Age, that hasn't happened. So normally the way it happens is as a point source exposure. So if you are a pregnant woman and you're smoking, that's affecting your DNA, right? So you're methylating it, you're turning genes off. So you're affecting, I'm not saying you're mutating the DNA, you're not changing the ATGs or Cs, well that too. But in an epigenetic sense, you're changing what genes have RNA transcribed, okay? So that smoke is affecting mom's gene. It's affecting the baby's DNA, okay? So the mom is F0, the baby is F1. And if the baby is a female, then it's affecting the baby's egg cells. So it's affecting the grandchild's DNA. All on that one day that the mom is smoking, that DNA is being affected, okay? Or alcohol might be a better example because it would be more systemic than smoke probably. I don't know that for the fetus actually, but that would be my guess. And I'll show you data on both of those for a single person. So if a mom is pregnant with a son, then you're only affecting two generations, F0 and F1, because you're not affecting the son's sperm, okay? Is that so? So this is a way in which, and we'll talk about father's epigenome effect too on the child, but long story short, moms are much more important because the baby's growing in them over a long period of time. And then if the baby is a daughter, her children, the grandchildren, can be affected, and men just don't have it. Okay. I usually do a show of hands on if you want me to cover this, and it's usually like 50-50. So I'm going to skip this. Totally not board testable. This isn't even me testable. But this is just showing C. elegans data that I think this was 14 generations. Yeah, they have an epigenetic effect 14 generations later. So there's in animal models we've seen it go past that F2, the 3 generations, but not really in humans much. That we've seen it in mice. I'll talk about that. But so stay tuned. But while people are kind of excited, like that would be crazy, because then people have talked, well, if that's the case, effects during slavery are still having ramifications now, right? And so it becomes a very societal thing you have to deal with on a much longer time frame. We definitely have to deal with 3 generations, so you still have to deal with over 100 years. But anyway, so in animal models, it seems to go, there's data of it going further. Okay, there's 2 slides. Any questions about what I've said so far? All right, there's 2 slides that students have the most problems with. This is one, and then kind of the clinical manifestation using the data from this is the other. So I'll explain it, and then I'll ask these questions. So this is showing how your epigenome gets reset, okay? So when the sperm comes in during fertilization, the sperm's DNA, which has an epigenetic signature, okay, right? Mine is, I run, I have alcohol, that's affected my DNA. That epigenetic signature is attacked by the egg. It's basically erased. So this y-axis is measuring global methylation. Remember the CG islands, and this is the easiest main way to talk about an epigenetic signature. So I have promoters that are methylated and turning those genes off. Well, that methylation is attacked upon fertilization and almost cleared out. You'll see it's not at 0, and that's going to become important. So there's something called imprinting, where there's some amount of methylation of the DNA that does not get erased initially during fertilization. So the sperm's epigenome is mostly erased. The female's actually is also erased to the same level, but in a different mechanism. Instead of it actively being the methyls being taken off, as the maternal DNA is copied, the epigenetic signature is not copied over. Okay, so if you copied a gene that had a methylated promoter, you could also have machinery that adds that methyl back on, but it is not in the egg. And so basically, there's not a clinical manifestation I know of that difference. The long and short of it is both of those, the egg and sperm DNA, both mostly get demethylated and kind of reset. And so this kind of gets back at like, how is it that we cannot live forever, but we can have, you know, children? Our species doesn't die out. And part of this is like, reflashing a hard drive, right? You are kind of making things as stem cellish as possible. And the epigenome is going to be used in development. So as things become, arms, legs, fingers, eyes, that's controlled through epigenetics. So these lines go down, okay, and so now we're looking at the DNA of this developing embryo, and they're reprogrammed for development, okay? And that changes as the cells kind of go into different organs and stuff, right? Okay, so I'm trying to do this slowly because it is a confusing slide. This really kind of could be two slides and it would be clearer. The part I just said on the left, that was one slide. This dashed line should kind of be a different slide in that now these lines, we're just talking about the cells in the germline. Okay, so we're just talking about what will become the egg and the sperm. Okay, so when I and Kathleen had our first child and second child, this happened, this first part, right? And then the programming, happened for the fetus to develop. When I had my daughter, okay, now I'll get into this later, but males pass on specific parts of the genome that is paternally imprinted, meaning there's sections that are methylated and shut off because it came from the dad, okay? And there are other sections from mom that are shut off because it came from mom, okay? And so that's why you can think down here, this non-going to zero on the left, that is the parts that were paternally or maternally imprinted that did not get erased, okay? Now, when I had my daughter, her cells will eventually need to be maternally imprinted, but mine were paternally imprinted. So in the germline, that even the maternal and paternal imprinting needs to be erased and reprogrammed according to the sex of the fetus, okay? Does that make sense to everyone? So my daughter's DNA that was paternally imprinted got erased and became maternally imprinted. So when she has a child and she comes in here, there's this maternal imprint here. We're going to have another slide on that that will maybe explain it more. So I'll pause after 3 more slides to see if it's clear. So you guys have heard about X inactivation. All women have 2 X's. We have 1 chromosome, X chromosome. 1 X chromosome in all women is epigenetically shut off, but it differs which X chromosome that is, and it even differs within one woman, right? So as a female fetus is developing, an X chromosome gets epigenetically shut off, and then when the cells are dividing, the other X chromosome could get shut off. It's kind of like a race when the cell creates a new, when divides and there's a new cell with 2 X chromosomes, one of them's chosen to be X inactivated. And so here they're color-coded according to which one's inactivated, and you can see in different tissues, there's different patterning. So like, and I don't know, again, a clinical, I don't know a clinical manifestation in a single spot, like this cell or this skin region, but there is a manifestation that cells will behave differently across organs and stuff, and so they could respond differently in cancers and that sort of thing. So anyway, so in the skin you see, like, I guess one cell divided and then it probably made these other cells that were all X inactivated the same way, but there was a striated pattern of red over there. Okay, so that's all you have to know about that. So let me back up. So this happens in animals, right? If you see color patterning in cats, for example, or it can be that was controlled epigenetically. Bees, when they need a queen bee, they actually feed the queen bee royal jelly, which creates the queen bee, changes the bee to be a queen bee. Okay, and so here on the right is a slide showing the male and female imprinting, okay? So I forget which is which. The gray, I think, is the, let's say the blue is the paternal imprinting and the gray is the maternal imprinting. I'm not going to require you to know any of these regions. What I do want to point out is that there's portions where, if what I said is right, is if blue is paternal imprinted, this part of the genome inherited from the father would be shut off. If this part of the genome was the copy that was inherited from the mom would have this version on, okay, this portion on. And then the main thing I want you to get is that there's regions where there's overlap, okay, where they can be shut off by both the mom and dad. And so we'll talk about a disease state with this, but what I want you to kind of think of this as is it's kind of like you have a mutation that shuts a gene off by default in these regions. You've already had one hit. It's not actually a mutation, it's just epigenetically off, but it does make you more prone to, I only have one gene to do this function, whatever's in there, and if I lose that, I've lost both, right? Okay. Here, about 3 more slides, and then I'll pause and ask for questions. So here's data showing how your epigenome changes in your time, in your life. And so there's like a lot of great twin studies. So we're looking at cytosine methylation again, and here's twins at 3 years old and at 50 years old, and you see they differ immensely. This heat map is, I think, of smokers, showing the difference over time. So just know that, you know, even in twins, it changes. It's very environment dependent, and so this lecture is definitely a nurture lecture as opposed to nature, right? Like nature obviously is our DNA, and so this is that, but this is showing that the environment, the nurture part, is affecting our biomedical health. Sorry, I think, is the X inactivation pattern why you see a mosaic effect in something like Turner syndrome? Turner syndrome, I would have to look up. I'll look up and answer you that. And sorry, there's another question. Is the active X chromosome the one that is contained within a released egg, or could it potentially be the silence copy too? Sorry, I'm not understanding that. Is the active X chromosome the one that is contained within the released egg? Sorry, so every cell has both X's in a female. One of them's just, oh, I see what you're saying. If you're having a, it depends, right? It depends. So if you had, if mom passed on her X to a son, and the father passed on a Y, then that X would have to be active. You're not going to inactivate that X. So it's only in females that one X is shut off. Okay, maybe 3 more slides till I stop. So normally, I think a lot of people, including students, think of whenever a gene is shut off, it's bad because we've evolved. However we evolved is what is perfect, right? But we're not like that, right? So there are bad things in us. You know, we have obesity now, so turning off genes can be good. It gets a lot easier when we define things as good or bad. So there's words we'll talk about like tumor suppressor. That is a good thing because it's suppressing tumors, that gene. So like P53 is a tumor suppressor. So turning off a tumor suppressor is bad. Sorry for the colorblind people, but these are color-coded good and bad. And so a proto-oncogene is bad, right? Because it can become a cancer. It can cause a cancer cell to form. And so turning off a proto-oncogene is good. All right. So here we're starting to get into some of the studies that could not be done. You know, you can't set this study up, so you just look at what happened. And so this over-colic study follows a famine that happened around the turn of the century, around the 1900s. And what they found was there's this slow growth period of development when people are around 10 years old, so 8 to 12, and they seem to be kind of prone to have an effect, right? the environment can affect them a lot then. And so anyway, if you look at grandparents that were starved during this famine, it turns out that their progeny, the kids and grandkids, had a protective effect. They had less heart disease and they were not as prone to diabetes. And so this has been termed the thrifty hypothesis, and there's been epigenetics links found to this. So the kind of anthropomorphization of this is that, again, like Lamarck and the giraffe, is that our genome has adapted for what environment we are exposed to, even if it's a non-ideal environment. And so then if we're exposed to it again, we're better able to handle it. And so this is kind of the mechanism through which some of the new research on fasting being so good is probably part of it. Now, most of all that new research is really, most of it's talking about as a single person, not passing it on, but there is data showing that can be passed on to generations, okay? In that same time period, if you look, should say not starved. If you, on the right, if you looked at people that had access to food, so rich people that were not in the same place at the same time but were not starving, their grandchildren had were 4 times more likely to have diabetes, type 2 diabetes. And so, and for some reason in the starved portion, you found that link through the son, but not through the daughter, but then you saw it in both the granddaughter and grandson. So the ramifications of this are pretty good, large in a public policy sense. So like if you talk about Medicare for all, which I'm a huge proponent for, there's, or if you talk about, that would be good in this case. If you talk about, for example, taxing, people are taxing salt and taxing fat food or diet sodas. If you start taxing people for things that you're kind of saying, this is your fault for being, it's your fault for being obese and costing the health care industry money, so taxes shouldn't pay for that. If you're taxing it somehow, it's very frowned upon if you're taxing people for something that's controlled genetically for them. So this is clearly a mixed effect, but if there's a 4-fold increased chance of them having diabetes because their parents ate a lot, their grandparents ate a lot, that does not seem just to tax them for the full amount of that diabetes. All right. So you guys will use BMI because everyone does. This is my slide to point out that I hate BMI. But it's what we have. So whenever people come in, you get their weight and you have their height, so you get the BMI. But if you had an Olympic weightlifter, they would seem obese because they have a lot of muscle, but they're not unhealthy. So here's a list of other things you could do that are better, right? There's these body shape indexes calculators. There's percent fat monitors. Audio is choppy and cutting out. Okay. I will try to change it back to the one that was bad before and see how that goes. All right. Let me know if that is better or worse. Was there anything I was talking about that you didn't hear that you want me to say again? I'm going to go over the imprinting again when I get to the disease state we're talking about with it. Okay. So here's another famine, Dutch famine, 1944 to 45. I believe the Nazis were pulling back and they destroyed the crops. So if you look at mothers that were pregnant and you look how their children and grandchildren did, it depends on kind of what trimester they were in. If you look at pregnant women in the first trimester, their children appeared to be okay, which kind of makes sense. And then if you look at the second or third trimester, the children became small for gestational age, SGA. And all of those make sense to me in that I think of the fetus kind of as a parasite, and if the mom is not having enough nutrition, then the baby, especially later on when it needs a lot of resources, will be small for gestational age. Nothing, that's super obvious. If you look at the grandchildren, the women that were in the first trimester had grandchildren that were small for gestational age. So again, actually, I don't know if that was just females or males, right? It would make more sense to just be females there, but I believe it was measuring females and males. So I don't know how that would be, or if that was just averaged out among the sexes. And then that kind of effect had gone away for the second and third trimester grandchildren. And when they went back and looked at the first trimester women, children, they found that there was a decrease in IGF-2 methylation 6 decades later. So the children that were in the mother in the first trimester, when she did not have resources, 6 decades later, they still had methylation, decreased methylation on a promoter. Question, Solomon?
Yeah, so with that, I'm a little confused because we talked about during fertilization, you have imprinting and then that kind of resets it. So why do we see SGA in the grandchildren if they're having a reset?
Yeah, so the reset is happening very, very early, right? The reset's happening when the sperm gets into the egg, right? But then if the mom was fasting for one day, you probably wouldn't see that. But this was a famine, so she was fasted for the entire pregnancy, right? So imagine after the reset, having, starting to put on an epigenetic signature for the embryo to develop. Well, during that time, just like if the mom was drinking alcohol, there's extra hits that to the fetus's epigenome. Those hits are malnutrition and being energy deprived. Is that good? I have another question. When a gene is maternally imprinted, is it paternally expressed? When I get to the second confusing slide, I will hopefully confuse you and then educate you, and then it will make sense. Lindsay, chime in after that slide and let me know if it's clear. And here's that slide. Okay. This is by far the slide that students have the most problems with. So whenever we have the virtual office hours, I'll probably work through this because usually students come up to my office and on the whiteboard we can work through it and it just makes a lot more sense. I've tried to make this slide similar to what I do on my whiteboard, hence how it's busy. Okay, so there are these two diseases, Prader-Willi syndrome and Angelman syndrome, and they both occur in a similar region of the genome, okay, this region right here. And so that region can be deleted in its entirety, okay, that sometimes happens. And so if, this doesn't happen, if you had that entire region deleted on both copies, you would have both of these. You would have Prader-Willi syndrome and Angelman syndrome. That doesn't happen, right? It's not a double deletion. So Prader-Willi, follow the P's that think Prader-Willi goes with paternal, meaning the paternal chromosome has the active Prader-Willi gene, which is good, meaning you don't have Prader-Willi. You have these diseases if the gene in that region is deleted, not functional, epigenetically off, if it's not working. So I passed on to my son and daughter awesome Prader-Willi sequence, okay? And it's working, so they do not have Prader-Willi, okay? And so that's shown down here in the green. So there's these green boxes, the top two, that are the Prader-Willi syndrome PWS region. And I passed on the green region, so those are normal DNA, actively transcribed, and my children don't have Prader-Willi. Tafleen, despite her best efforts, passed on, and every woman passes on epigenetically inactivated Prader-Willi syndrome regions, okay? So there's two red boxes right here. So I have the X's here, okay? So if my children had a deletion of this region from me, okay, they would have Prader-Willi because the DNA from Tafleen is epigenetically off. And now they don't have the functional DNA from me because it was deleted, okay? Angelman syndrome, the only thing I could think of there is that your mom's an angel. So if the mom passes on functional Angelman syndrome, right? Tafleen's Angelman syndrome gene down here, AS gene green, is awesome. Our kids don't have Angelman syndrome. I, despite my best efforts and every man, passes on epigenetically off Angelman syndrome gene, okay? So if there was a deletion in the chromosome that Tafleen passed on from the mom of this region, my children would have Angelman syndrome because they lost the Angelman syndrome gene that was active. Does that make sense to everyone? This is the imprinted region that did not get erased upon fertilization, right? Because I have paternal imprinting, my Angelman syndrome gene was paternally imprinted off. And the mom's DNA is paternally imprinted to have Prader-Willi syndrome region off. The imprinting is defined as where you have methylation that shuts a gene off that lasts that first wave of erasing the epigenetic signature. So does it make it autosomal dominant even though two regions that are off are required for the disease? It is, if you're talking about getting that deletion from a specific sex, so if you're talking about getting the deletion from me, then that one deletion was enough for my children to have Prader-Willi, right? But I think I'm going to answer a poll everywhere by answering this question. Hold on, let me read the second question. Hypothetically, if you can unmethylate parental Angelman gene, would the issue be resolved? Yes. Yeah, that is a potential therapeutic that we don't functionally have. So let's say my genome was deleted in my children in this Prader-Willi syndrome and Angelman syndrome gene. Okay, when you delete that from what I passed on, deleting the Angelman syndrome gene didn't matter at all because it was already epigenetically off. Deleting the Prader-Willi syndrome gene did matter because it was epigenetically on. Now, and normally these children don't actually have children because there's the sperm in one is immotile and there's significant retardation in the other. But if they did have children, it would depend if they're a male or female on what they pass on, right? So let's say I luckily have a daughter and a son, so I can always use them in this lecture. Let's say they both had a deletion from me and they both had Prader-Willi syndrome gene, or Prader-Willi syndrome, the disease, because they had got the deletion from me, right? If they both somehow had a kid, my son would either pass on the deleted chromosome from me or the chromosome from Tafleen that's not deleted, but it has been re-imprinted male because he is male. And so he turned off the Prader-Willi gene, right? So he would be, either way, what he passes on is kind of similar in Prader-Willi syndrome sense because they're going to be off either way. There is a difference on what he passes on regarding, actually, no, yeah, it just seems similar. But if my daughter were passing something on, if she passed on the a deletion, again, it's just not there. If she passed on the non-deleted region, hers would be reprogrammed with Angelman syndrome on. Okay, so, okay, yep, Tasia, question.
So the male child would pass it on regardless if he had Prader-Willi syndrome. Then wouldn't that mean like every guy could pass it on like 50% then or something, or would it just be
no? So like on? Okay, so if we talk about all of us, right? What is in all of us? All of us don't have this deletion in either region, in either chromosome, right? So all of us have Prader-Willi that's functional from our dad, and we have Angelman syndrome that's functional from our mom, okay? If you pass on a deletion, a gene deletion, then if the mom or dad passes on that deletion, it changes what the child will have because, and you just think about what the other sex is passing on. So if a male passes on a deletion, the child will not have a functional Prader-Willi, right? And so they will have Prader-Willi syndrome. Does that make sense?
Yeah, but like the one they got from the mom would, if they passed it on to their child, they would turn on that Prader-Willi gene if they had a functional copy of it. That was
well. Whatever you get from your parents is reprogrammed to be your sex for your child, but not for you, right? So, if this was deleted, and I'll just work through a son and daughter. If this was deleted and my son had Prader-Willi, right? Because I deleted the greens, okay? Well, because so he's still male. So he has Prader-Willi. When he has a child, which again doesn't really happen, but it's a good question. If he had, and you could, for example, these people could have children if, remember I said the sperm was immotile, if you did like in vitro and took the sperm out, the sperm's functional, so it could work that way. If he had a child, if he passes on the functional chromosome that came from mom, that would have been reprogrammed like normal male patterning. So he would be passing on a functional Prader-Willi region, even though it came from mom because it was re-imprinted male. Okay, that's the kind of thing that students get hooked up on, that the mom passed on a functional Angelman syndrome and not Prader-Willi, which happened to the child. But if the child's male, his sperm will reprogram that to be functional Prader-Willi and not functional Angelman syndrome. And it's the reverse for a female, right? So the daughter wouldn't, again, if you don't have the deletion, you don't have the deletion. But if my daughter had Prader-Willi, because this was deleted, and then she passed on the functional chromosome, the functional chromosome came from mom, and it's going to be still programmed like a female because she's a daughter. So then she would be passing on functional Angelman syndrome and not Prader-Willi. Or sorry, unfunctional Prader-Willi, right?
Okay, so, thank you. That makes sense.
And so another wrench to throw in here is that you can have something called uniparental disomy, where both of the chromosomes can come from one parent. Okay, and this happens not an insignificant. It's not the majority of the time, but it might be like a quarter of the time or something. And I don't know what fraction it is, but it does happen. So if that happens, there doesn't have to be a deletion. So if for some reason my children in this region got both chromosome regions from me, even if there were no deletions, what disease would they have? Someone type that into the chat. That's right. They would have Angelman because I'm passing off two imprinted off Angelman syndrome genes. Okay. And so I don't know if you guys, I hope you're using your anatomy groups or other groups to do studying in groups somehow, because it's very useful. I highly recommend that you make up quiz questions for this and like throw in, okay, I gave you one example, I think here. And if I might have the answer at the end of the PowerPoint, and if not, I'll work through this one in the virtual office hours. Okay. All right. Let me hit one more slide and then I'll pause for questions. So this is just a funny real world thing that I snapped a picture of. When Kathleen and I were having our first child, we had genetics counseling and we got this flyer. We're sitting in the doctor's office and they gave us this flyer. And first of all, the genetic counselor got the number of chromosomes wrong. They didn't, remember the company 23andMe, right? So you get 23 from each parent. They named an odd number that was close to double that, but wrong, which is wrong for a bunch of reasons. So anyway, so there's on this Angelman and Prader-Willi. So I asked the doctor, I was like, oh, is this testing for epigenetic effects? Because you can just have deletions, you can have point mutations, or you could have this, epigenetically off. And long story short, deer in the headlights, they had no idea what I was talking about. And so I ended up calling the company and eventually got through, and they admitted to me that, one, it's not testing for any epigenetics. And then if they find that it might be Angelman versus Prader-Willi, they send it to another company to figure out what's going on. And then they said to me, but that never happens because no one's ever positive. So the long story short is, if you're in a clinic and you're pitching something, know what you're pitching. Also, they didn't have Fragile X, which I found interesting. Okay, so we're going to move on from that. Are there any questions about that section? All right, let's go for 5 more minutes and then we'll stop. This is another slide. You can just put a big X out. I will not test you on it, and the boards definitely won't, but I found it very interesting. Here's an example of a gene that's been found in the last 4 or 5 years, that TRIM28, that's been found to be linked to obesity. So we have this global obesity epidemic, but we could not have evolved as quickly as the obesity epidemics come on. So it's clearly not DNA mutation changes, ATGCs. And so long story short, if you look at the methylation of TRIM, you don't see a bell curve. You see that there's this big bump where obese people have this epigenetic modification, and TRIM's involved in that. So long story short, again, this is another thing where it's hard to, it comes into play with what do you want to tax. So here are lean people in blue, and we're looking at the TRIM mRNA expression. So lean people are 31%. The fraction that is epigenetically shut off is different in obese people, basically. Since it's an FYI, we'll move on from that. All right. So again, a big take home for cancer is that you can have these kind of gatekeeper drugs that prevent you from getting more mutations and problems, or that can take care of cells if they're getting out of control, like p53. And if they get hypermethylated, which shuts them off, that can lead to cancer. So in breast cancer, 13% have hypermethylation of the BRCA1 gene, and in colorectal cancer, there's hypermethylation of this MGMT gene, which is involved in DNA repair, and that's like over 50%. Very high. Sorry, I'm looking at the chats. Okay. And so here is a table showing you that after the initial hypermethylation, shutting a gene off, then you have mass hypomethylation. So there's a gatekeeper that's preventing things from going bad. That is methylated in the promoter region and shut off. And then a lot of genes get hypomethylated and turned on. And so these are different cancers that have global hypomethylation later. Cancer in the tissue, blood, aging, environmental exposures, cardiovascular disease. And so we talked about varinostat before and how it's involved. We already had this slide, so I'll move on to that as an epigenetic drug. And so we'll take a break here, and in the next half, I'm going to go over kind of all these things that we do in our life and the data behind how much they change our epigenome and what we can do: caloric restriction, exercise, sleep, inflammation, trauma. Let's see. I'd say we should. Yeah, let's take a. Okay, I'll on the fly in the chat make up this. Say if you want to keep, how many minutes of a break do you want? 0, 5, or 10? We could keep going with 0 minutes, do 5 or 10. All right, it's between 5 and 10. So let's come back in 7 minutes at 11:07. Okay. 7.5, I like it. All right, see you at 11:07.
Dr. Arbor, can I ask you a question about that last lecture?
Yeah, hold on. Let me stop the Panopto recording.